Hi, I'm Isabelle Breier, and I'll be discussing my research on regulatory elements of the gene engrailed1. All homeotherms, or warm-blooded animals, face an internally biological rather than environmental challenge of thermoregulation. Most mammals have thick hair or fur on their bodies and sweat glands only on the bottoms of their feet, and it should be noted that they use the glands for traction rather than thermoregulation. But humans have a unique adaptive trait for thermoregulation. We have a high density of sweat glands all over our bodies, as well as a small hair follicle size. We have both sweat and a large skin surface area from which the sweat can evaporate and cool us. We also lack substantial hair cover, which would impede evaporation. Our increase in sweat gland density and reduction in hair follicle size, derived traits that are clearly not present in our closest living chimpanzee relatives, are adaptations that likely evolved in our hominin ancestors. These traits likely facilitated long-distance bipedal locomotion in the genus Australopithecus and were likely critical for the evolution of endurance running capabilities in our genus of Homo. Interestingly, these different phenotypes are all controlled by the same gene, and it is the differential expression of this gene that likely modifies the phenotypes. Previous research has shown that expression of the gene engrailed1, abbreviated as EN1, is important in regulating the development of sweat glands and hair follicles. This picture shows an EN1 transgenic mouse, or mouse that has been genetically engineered to overexpress EN1, only in the basal layer of the skin, when the drug doxycycline is injected. The graph on the left demonstrates that the loss of EN1 expression in mice causes a loss of sweat or eccrine glands, while increasing EN1 levels cause a gain in sweat glands. The doxycycline-injected mouse at the right in the picture clearly has a thinner coat. This effect is quantified in the graph on the left. This phenotype is not due to a loss of hair, but rather to a smaller size of hair. Its hair is on average 3 cells thick instead of 4 cells thick. In sum, the mouse overexpressing EN1 has more sweat glands and smaller hairs. In humans, EN1 is also expressed at the time of sweat gland formation. Therefore, we hypothesize that mutations which led to an increase in EN1 expression have contributed to the human skin phenotype of increased sweat gland density and smaller hairs. However, we don't know what regulates EN1 expression. Previous work has provided a clue in showing that EN1 expression in skin is highly conserved throughout species, or similar throughout many species. The microscope image on the left shows a chicken limb that has been stained using an in situ hybridization assay, which shows where a gene is being expressed, so the dark blue to purple colored spots are where EN1 is being upregulated or highly expressed. In the case of the chickens, the upregulation causes the formation of ventral scales. The picture on the right shows the murine analog of this story. The purple areas of an in situ assay belie EN1 expression only in sweat gland primordia. Since EN1 expression is also conserved in humans, albeit with mutations that produce our unique phenotype, at least part of the regulatory mechanism must be conserved, but we don't know where that mechanism is. Since EN1 expression in skin is so highly conserved, at least part of the regulatory mechanism for the gene must also be conserved within vertebrates. We know that most evolutionarily relevant mutations lie in regulatory elements of genes, not in coding portions. There must be some sequence constraint and some ancient enhancers that we share. To find out, we delved deep into the most likely place we'd find these regulatory elements, in EN1's topologically associated domain, or TAD. A TAD is the region around a certain gene containing a high level of interaction between genes. Our hypothesis about the conservation within EN1's TADs turned out to be true. As this diagram shows, regions of the genome align for many species. Each blue line represents a 100 base pair long evolutionary conserved region, or ECR for short, which is a sequence that is nearly the same in many distantly related species, indicating that they code for traits with basic and vital functions. When the sequence is this conserved, it suggests that a functional constraint is at play, so these candidate ECRs may be acting as enhancers of EN1, or short regions of DNA that increase the likelihood that the gene will be expressed. However, it is unknown how they are regulated. All we know is that the regulatory elements controlling EN1 expression, whatever they may be, mutated over the course of human evolution and contributed to the high sweat gland density and small hair follicle size that is distinctive of our species. Therefore, the goal of this project was to define the regulatory elements that control EN1 expression. We first designed primers for each ECR using IDT software. The primers allow the ECR of interest to be expressed. Then we utilized polymerase chain reaction, or PCR, to efficiently produce millions of copies of the primer. We then cloned the ECR using lentiviruses as a vector. This involves splicing the ECR sequence, flanked by its forward primer in front and reverse primer at the end, into the lentivirus's genome. The lentivirus then expressed our genetic material of interest and created many copies of it. We then sequenced the lentivirus genome to ensure that the gene inserted properly. After making sure it had, we injected mouse embryos with the gene 9 days after conception. The mice gestated, were born, and lived 2.5 days postnatally, at which point we sacrificed them. This is a key moment for observing their placodes, or primordial structures that will later develop into sweat glands. We harvested their feet and sliced them into 12-micron thick sections, at which point we immunostained them. We used a brownish stain called DAB to highlight the expression of the ECR in question. We used a fluorescent red stain to highlight the protein keratin 14, which is a marker of the cells in the deepest layer of the skin in which EN1 is expressed. Staining for keratin 14 allows us to ascertain that the enhancer is active in the right cell type. We also applied a stain called DAPI, which colors nuclei blue. We found that both the mouse and human sequences of ECR15 are active regulators of EN1 expression. As you can see in the DAB staining images at the top, the dark brown color of the placode areas demonstrates that both species analogs of ECR15 are highly expressed. In other words, ECR15 serves as an enhancer upregulating the expression of EN1 in sweat gland primordia. The red and blue images show the DAPI and keratin 14 staining in the same feet. The red areas demonstrate that keratin 14 is highly expressed in the same areas as ECR15. As you may recall, the bottom image from a previous publication displays EN1 expression in a mouse. I have included it for comparison because it demonstrates that ECR15 is active in the same tissues where EN1 is expressed. In conclusion, our human sweat gland and hair follicle phenotype likely facilitated the evolution of our long-distance bipedal locomotion abilities. EN1 controls sweat gland and hair follicle phenotypes, and as demonstrated by ECR screening, mutations in ECR15 as a regulator of EN1 likely contributed to our unique phenotype. In the future, we will screen the remaining ECRs. In fact, it is likely that multiple elements control EN1 expression. ECR7 is a potentially promising candidate. We will also test mouse and chimp versions of ECRs. We will also breed knockout mice, or ones with deletions of various elements, to test the deleted element's functional significance. We also intend to examine how much the human EN1 gene and regulatory mechanism diverge from those of other species and determine the functional significance of those divergences. In addition, when disease or injuries like burns hamper these physiological mechanisms, there is significant clinical morbidity. A future application of this research may lie in the regeneration of skin with normal densities of sweat glands and normally small hair follicles. I'd like to acknowledge my lab, our collaborators, and of course the BFS program. Thank you for listening.